Alright guys, so today is our second proteins lecture, and we're going to use this time to talk about the acid-base properties of amino acids. So amino acids have both a carboxyl group and an amino group. So that means they can act as a weak acid with the carboxylic acid group, or a weak base with the amino group. So what we're going to do is on the board, we're going to draw the acid dissociation equation for alanine, the base dissociation equation for alanine, and then we're going to combine both of the equations. So what I want you to do first is look back at your notes and write down the general equation for an acid in water and the general equation for a base in water. And then I want you to look up the structure of alanine for me, the amino acid alanine. So either look at page 460 in your book or our first proteins lecture or the amino acids handout on Blackboard. Okay, so an acid in water. So we have HA is our acid. We put it in water. So in this case, HA, the acid, is going to act as a proton donor because it's an acid. So that means water has to act as a base here. So HA is going to lose that proton. So we have A minus, and it's going to donate it to the water, giving us a hydronium ion. For a base, B, when we react that with water, the base is going to be our proton acceptor. So that means water has to act as the acid, the proton donor. So we end up with BH plus and a hydroxide ion. Okay, now let's substitute HA and B for alanine. Okay, so alanine has just a methyl group for a side chain. So there's our amino group. Here's our alpha carbon, our carbonyl carbon, and then here is our methyl group side chain. We're going to react it with water. We're going to place this alanine into water, and just like the equation above, we're going to donate a proton to water. So I want you to pause the video and see if you can tell me where this proton is going to come from. So what part of alanine can act as an acid? Okay, so the carboxylic acid is the part of alanine that can act as an acid. So we're going to donate a proton from that carboxyl group. So we have a negative charge on our alanine here. And we still form hydronium ion. Okay, now let's draw alanine again. And this time we're going to have alanine act as the base. So I want you to go ahead and pause the video and tell me which part of alanine can act as a base. So it is the amino group, because we learned at the beginning of the semester that amines can act as weak bases. So our NH2 group is going to be able to accept a proton from water. Okay, so when we have an amino acid and we add it to water, how do we know if it's going to act as an acid or base? Well, the answer is both. So combining the two lower equations into one reaction, we're going to perform two acid-base neutralizations. We're going to do an acid-base neutralization between the amino group and one water molecule, and an acid-base neutralization between the carboxylic acid group and one water molecule. So we're going to end up with a positively charged nitrogen and a negative charge on our oxygen, a hydronium ion and a hydroxide ion. And then the hydronium ion and the hydroxide ion can come together to regenerate our two water molecules. So what we've created here is what we call a zwitterion. So zwitterions are molecules with two or more functional groups, at least one positively charged and at least one negatively charged. So when drawing amino acids, I want you to draw them as zwitterions unless told otherwise, but a fair bit of warning. These are pH dependent. So what that means is there's a certain pH range where amino acids meet this definition, where they're going to have both a positively charged group and a negatively charged group. So let's talk about that a little bit more. So the isoelectric point of a molecule is going to be the pH at which a molecule has a charge of 0. And we're going to calculate the pi of amino acid with these steps right here. I know this looks like a bunch, but we're going to go through it together on the board with alanine and lysine. So we're going to start by drawing the amino acid at pH 1. Then we're going to label each titratable group with the pKa. We're going to move up from pH 1 until we reach the first pKa. We're going to draw a reversible reaction arrow and write the lowest pKa value on top of the arrow. And then we're going to draw a new structure with the correct proton removed. We're going to repeat this until we remove every proton that can be removed. Then we're going to identify the net charge for each species you drew. So then we're going to find the species with 0 net charge. And our pi is going to be the average of the pKa you wrote above the reaction arrow on both sides of the neutral species. Okay, so this will make a lot more sense when we actually work through an example. So let's go back to the board and let's try this with alanine. So first thing you got to ask yourself is, what does alanine look like at pH 1? So at pH 1 is a super acidic environment. So anywhere a proton can bind, a proton will bind. So that means our amino group is going to have a positive charge because a proton can bind there. And our carboxylic acid group is going to have a proton bound there, so it's going to be neutrally charged. So I want you to go ahead and pause this video and look at your amino acid handout and tell me what the pKa values are for alanine. So you should see 2 pKa values listed for alanine. You should see a pKaC and a pKaN. So what that means is the pKaC is for our carboxyl group, and the pKaN is for our amino group. So the pKaC looks like it is 2.34, and the pKaN looks like 9.69. And we are currently at pH 1. So the next thing we're going to do is put our lowest pKa above the reaction arrow. So 2.34. So as we move up in pH, as we start approaching this pKa of 2.34 here, we're going to start deprotonating this carboxylic acid. And when we get to a pH of exactly 2.34, that means that we're going to have an equal mix of the carboxylic acid form and the carboxylate ion form. So as we move way past pH 2.34, we're going to completely deprotonate that carboxylic acid group. And so our alanine is going to look like this right here. Okay, so what is our next pKa we're going to reach? So our next pKa should be 9.69. And that corresponds to this amino group. So as we blow past 9.69 in pH, as we get well above 9.69, we're going to completely deprotonate this azanium group. And so we should get a species that looks like this right here. Okay, so now what I want you to do is pause the video and tell me what the net charge is on each of these 3 species we drew. Okay, so this first one should be a positive 1 because we have a single positive charge. The second one should be neutral, should be 0 because we have a positive 1 and a -1. Add those together, we get 0. And this last one, all we have is a single negative charge, so the net charge will be -1. So that means our neutral species is the one in the middle. And so we're going to calculate the pi as the average of the pKa's on both sides of this neutral species. So that means our pi is going to be equal to 2.34 plus 9.69 divided by 2. So go ahead and pause the video and see if you can tell me what that is. So it looks like it is 6.015. Okay, so now what I want you to do is see if you can do this for the amino acid lysine. See if you can repeat this whole process on your own and then watch the next video and we'll work through that example together.